Protein phosphorylation is also regulated by proteasomes. The precise dependence between protein phosphorylation and microtubule sliding and movement initiation is still under investigation. A Ca2+ mediated and or CAMP-dependent phosphorylation signaling mechanism through the radial spoke/central pair system of the axoneme has been proposed. The potential role of reactive oxygen species, ROS, generated at the contact of sperm with aerobic conditions such as the external medium at ejaculation and the molecular mechanisms by which these reactive metabolites exert their biological activity has been put forward by Baker and Aiken. A gas, NO, was also observed to enhance motility of fathead minnow spermatozoa. Nevertheless, the mechanism by which NO affects sperm motility is probably unrelated to osmolality, because of its very low active concentration range. During the period after spermatozoa comes in contact with an activating medium, the ion concentrations inside the sperm cell are rebalanced, and osmotic pressure affecting the sperm membrane becomes harmful for sperm integrity, limiting the period of motility to a short interval. These phenomena are much faster and more obvious in freshwater species, in which sperm motility usually does not last for more than 0.5 to 2 minutes. Video 1 can be viewed at https://vimeo.com/310085381. Brook trout, Salvelinus fontinalis, spermatozoa recorded while swimming in a swimming medium composed of low osmolarity, 10 tris at pH 8.0 plus 10 Ca-Cl2. Remark the briefness of motility, around 30 s duration at 10 degrees Celsius, courtesy of Dr. Galina Prokopchuk. In case of sperm collection for artificial reproduction or in vitro studies, particular care should be taken to avoid precocious activation of motility. One should avoid any contact of sperm cells with external water or urine during stripping. Any assessment of motility parameters should be started as soon as possible after sperm activation, bearing in mind that the earliest period of motility, the most efficient one, should be characterized. Fish spermatozoa are usually characterized by a very high initial velocity, up to 200 ms-1, due to the high flagellar beat frequency, up to 100 Hz. This most active period lasts only a few seconds immediately after contact with the activating medium. Values of all motility parameters decrease rapidly immediately after initiation of fish sperm flagellum movement, which is why any motility parameter must refer to a precise time point after activation for intra or interspecies comparisons. Generally, the duration of motility is a trade-off between the level of energy stocks possessed by a cell and the process of osmotic damage experienced by the cell. The latter is more critical in freshwater fish species, and the former is important for marine fish. At a precise time point, most spermatozoa have very similar characteristics. Video 2 can be viewed at https://vimeo.com/310086859. Carp, Cyprinus carpio, spermatozoa recorded while swimming in 2 different media successively. The first part of the record shows the carp sperm population activated in a swimming solution composed of 45 NaCl plus 5 KCl plus tris at pH 8.0, while the second part shows spermatozoan activation in a low osmolarity medium, distilled water plus tris at pH 8.0. Remark the curling of the flagellar tip which limits the duration of motility at low osmolarity, courtesy of Dr. Volodymyr Bondarenko.